joining event shown on the left side of this slide that can sometimes introduce a small disruption to the DNA sequence. Or there can be an integration of DNA that has homology to the sequence of DNA flanking the double-stranded break. And in that case, a new piece of genetic information is incorporated into the genome at the site of the original break. This was research that had been done over the previous couple of decades before Emmanuel and I did our work with Cas9, but we recognized that the activity of Cas9, its ability to introduce a double-stranded break to DNA at a desired position by directing it with these single guide RNAs, could allow scientists to introduce double-stranded breaks into a genome that could trigger the kind of repair that I'm showing here. And so we imagined a system working very much like in this video, where the Cas9 protein could be directed to enter the nucleus of a eukaryotic cell, and with its guide RNA, search the genome for a 20 base pair sequence that would match the 20 nucleotides, the 20 letters of the guide RNA that would provide a match. And when that match occurs, we now understand that Cas9 is able to unwind the DNA. It allows the Cas9 protein to generate a precise double-stranded break in the DNA by cleaving each strand of DNA. And then those broken ends are handed off to repair enzymes in eukaryotic cells that lead to DNA repair. And in the process of repairing the DNA, there is the ability to introduce a change to the genome at a precise place, which is really the definition of genome engineering. So on the next few slides, I want to share a few things that we've learned over the last few years about how Cas9 is able to achieve this kind of editing in genomes of cells by triggering double-stranded breaks. And I'll start with showing a molecular model of the Cas9 protein. And this is based on a crystallographic structure that was solved originally by several different laboratories, our own, the lab of Martin Jinek, the lab of Osamu Nurecki, and subsequently many others who have contributed to understanding the actual molecular basis for Cas9's function. And in this slide, what you can see is the white protein, Cas9, holding on to its orange guide RNA and a blue double helical DNA molecule. And I'd like to point out that in this structure, we can see the mechanism by which Cas9 uses an RNA guide to interact with DNA at a precise position, because you can see the orange and blue helix formed inside the Cas9 protein representing the interaction between the guide RNA and one strand of the target DNA molecule. This is the actual way that these proteins are able to find and hold on to DNA prior to DNA cleavage. The next thing that we learned in the lab in studying how this cleavage event actually works is that a series of experiments showed that Cas9 is a highly dynamic protein. It has to be able to handle DNA, unwind a double helix the way we know it does. It has to be able to move. And through a series of chemical experiments that allowed us to detect motions in different parts of the protein, we discovered that Cas9 undergoes a large conformational change as it holds on to DNA and catalyzes cutting. And I'll show that in this video here. So this shows the Cas9 protein alone morphing to the structure it forms when it binds to the guide RNA, which you can see now in orange. This is the structure that is able to search the cell looking for a matching sequence of DNA. And when that match occurs, there's an additional structural change in the protein that accommodates the DNA molecule, so that you can now see the RNA-DNA helix forming inside of Cas9.
你真的要是特别想考上专升本，你就记住一点，就是你考试的前一天，你都保持一个什么心态呢？就是我必须考上，我不能考不上。 就是你不要给自己留任何的退路，我就是这么上去的，你就记住这一点就行了。因为我考试之前，就是我在备考期间，我也会上 B 站啊什么的，我就看一些那种经验呢，就怎么能考上专升本呢？专升本你要注意些什么呢？说实话，我看完之后呢，我也忘记了，因为我记性也不好。然后还有一点就是，我觉得那些学习技巧啊，什么比如说时间管理啊、学习方法呀，那都是别人的。我认为每个人都有自己的方法，就是你看那些其实没有什么用。首先，它不一定适合你。说实话，就算你没那些方法，你就是自己就有效率的去学习，你也能学到东西的。最大的原因是什么？是你不去学，你看完之后放刷牙吃灰，它有什么用呢？那我就认为没有用。说实话，我之前我也看那些视频，根本就没有用。但是我就是经过我的经验，观察周围，就是我们考上的人和那些没考上的人有一个什么特点啊？就是一定不要假学习。我是真的在学，虽然可能时间不多，但是我真的很有效果。那我有一个同学呢，他没考上，他就是上了个3本，他是怎么回事呢？就是他看起来比我要努力很多，但是呢，他每天到那之后，他就开始玩手机，别人在看着他玩手机，然后订外卖，然后玩手机，然后睡觉。但是呢，他确实在补课班待了一天，有什么用呢？或者就是就是做数学题，然后发朋友圈啊，我太努力了。我就说实话了，真的不难。你想想，我这底子我都能考上，而且我并没有付出全部的努力。我只能说，考不上的人有3种。第一种呢，就是他根本就不努力，他中途就放弃自己了。因为有些人呢，就是我努着力，然后放肆了几天。因为我如果放肆了几天，玩了几天，玩了一段时间，我也不会觉得，哎呀，我跟不上了，那我就放弃了吧？不会，我一直都是我考不上我就死去。虽然很偏激，但是我就一直就有这很强烈的想法，我并没有说我考不上之类的，我并没有给自己消极的想法。第二种就是假努力，就是努力给别人看，最后好像搞的就是说他已经那么努力了还是没考上，这也太难了吧？并不是，那种人他就是假学习给你们看的，自己他努不努力自己心里清楚。第3种他不希望你能考上，能明白吗？毕竟呢，你说专升本有没有难度？它肯定有一定的难度。它不难的话全考上了，你拿什么去甩掉别人呢？你要想，它有一定难度是好的，要不然大家都上去了。但是这个事情是什么呢？还好，大多数人都很懒惰，你只要勤奋一些，你就可以甩掉他们了，真的挺好的。所以你就记住这一点，绝对不要放弃。一定不要放弃，哪怕说玩了几天啊，我就觉得自己很愧疚啊，你得补就行了呀。我学习效率也非常的低，后来我就发现我学习效率低，我怎么做呢？我从早到晚我就坐那儿，它不低吗？我学1天，我看它能低到什么程度，我就拿时间来补。就是你要坚持住，我不在乎你学了多少，你学你就真学，你玩你就玩，你懂吗？你不要边学边玩，最后学也没学好，玩也没玩好。我认为呢，专升本最重要的根本就不是学习，你要学呢，大家都学。我觉得就是专升本一开始大家都有那股劲儿哈，就好像我必须要考上，我必须要赢，然后中途就是放弃了。其实你会发现，最后的赢家呀，最后胜利的人呢，都是因为坚持下来了。他可能付出的根本就有的时候那股劲儿啊，都没有别人在一开始的时候就是那么的用功，但是他可能就用一丢丢功，每天都用功，或者是说就算我3天打鱼2天晒网，我就学一点点，积累下来了，我到最后我都没放弃，他就赢了。